Good. You know, when you get old, one of the advantages you have is that you are often invited as the keynote or the distinguished professors. And I think it is just a matter of time before one becomes extinguished professor. So before that happens, I hope I'm able to do something important in what we've been doing since 1990, which is the mechanism of protein secretion. So I must also thank the very friendly organizers, and that's one thing you need in this business. You will develop a lot of people who probably don't like you, but it's good to have some good friends in high places because they make this kind of stuff happen. So thank you very much. Yeah. Suzhou is an amazing place, and this is my third trip to China in the last 2 years. And in fact, I'm planning to spend a lot of time here. So we can discuss all of that over a glass of beer, perhaps. So let me just give you, my talk is going to be more didactic. It's not going to be just filled with data. If you want to see data, we have published enough, and we'll continue to do so, and you can look for details in our publications. But I want to highlight how we got into this field, where we are, and where we think we might be going in the next 5 to 6 years. So I work on protein secretion, the conventional pathway. We call it conventional only because it was the first one defined. So the conventional pathway defined by George Palade, I mean, he did his work in the 60s and the 50s, and he was awarded the Nobel Prize in 1974. And if you look at his Nobel lecture, this is the drawing from his Nobel lecture, it's rather crude. He basically said that proteins enter the endoplasmic reticulum. Those proteins that are to be secreted, they go from ER to the Golgi. They can be seen in large granules and then outside of the cell. And this became the basic principle of vesicle-mediated protein secretion. Very important because this allows a cell to release material that needs to be released without ever changing the organization of the compartments where the protein is made or it's trafficked through, right? So the ER remains ER, Golgi remains Golgi, and yet you can release material to the outside of the cell. So since 1974, then great minds, Rothman, Sheckman, and Tom Sudoff, and members of their laboratories, spent almost 50 years trying to understand how you create a vesicle and how you collect material into a vesicle and then target the vesicle to the respective destination for it to fuse and so on and so forth. So, you know, we were doing that. So we were studying how you secrete proteins, how you make a Golgi. In fact, our latest interest is on how you secrete big molecules like the collagens and the mucins, which cannot be explained simply by the small vesicle-mediated transport that had been identified by the luminaries here. Yeah? So in addition to that, one of my favorite topics in the context of a cell has been, how does a cell create a structure like the Golgi apparatus? To be quite honest with you, we still don't really know where the Golgi begins and where it ends after years and years of working on this topic. And there's a lot of controversy. And we were interested in trying to understand, how is it that a Golgi complex made up of anything from 4 to 30 or 40 cisternae, as in the case of plants, how do you stack these cisternae? And Graham Warren and colleagues had identified proteins called grasps. Yeah? They had demonstrated, based on in vitro biology, that grasps are the molecules that stick or glue these cisternae in this orderly manner. Now, when the paper was published, there was an instantaneous alarm because yeast contain grasp but do not have a stacked cisterna of Golgi apparatus. Plants contain beautifully stacked Golgi cisternae, but don't contain grasps. So how do you explain that? And so, you know, lots of arguments and experiments were done claiming, yeah, in vitro versus in vivo, or there must be an alternative mechanism. All of that is correct. But we wanted to address this issue at the molecular level. So we finally deleted the GRASP gene. There are two in mammalian system, GRASP 55 and 65, and you can remove both of those from the mouse, and the mouse is more or less fine. Yeah? And even more importantly, there is not really a tremendous change in the organization of the Golgi complex. This was a bit of a blow. I'm sure there is some role in stacking, some aspect of it, but overall, the cell doesn't really care whether the cell contains GRASP proteins or not. Yeah? So then what exactly is the function of this protein? Now, we decided, well, before this work was published, we had decided in the early 2000s that we would try to delete the GRASP gene. There's only single gene in lower eukaryotes. So we decided to delete the GRASP gene from from the Dictyostelium discoideum, slime molds. Okay? They have only one, so the mammalians have two, mammals have two, whereas lower eukaryotes, including yeast, has only one. So just to give you a little bit of an understanding of how Dictyostelium lifespan works, under conditions of nutrient growth, it's a single cell organism. When you starve them, meaning you remove the source of carbon and nitrogen, then this cell undergoes a change in developmental steps in creating two different kinds of cell types. One called the stalk cells, and another one that produces the spores. And these will remain as they are shown here under conditions of starvation. The moment you put these cells back in nutrient-rich environment, then the spores are viable and they go back into this state again. Yeah? So we deleted this GRPA, that's the name of the gene in Dictyostelium. And lo and behold, we found no effect under conditions of growth. That was a bit of a blow. So we were desperately trying to find a physiological role for this particular gene. So under conditions of growth, there was no phenotype. Now, the point to remember is that when these cells are starved, the pre-spore cells secrete a protein called acyl-CoA binding activity, yeah? acbA. It is secreted, then upon secretion, pre-stalk cells carrying a protease called TAG-C cleave this acbA into a small peptide called spore differentiation factor number 2. Yeah? This works back on these cells via the AMPK pathway to produce more acbA and other components which keep this spore viable. Okay? If you do not have SDF2, then this spore is not viable. What Matt Kinseth in my lab found that cells lacking the GRPA GRASP gene produce spores, but they're not viable. Only 20 to 25% of the spores are viable. And we had a very crude bioassay which allowed us to quantitate the amount of SDF produced by these cells. And there was only less than 0.2 units compared to 10,000 units produced by the wild type. So this gave us a clue that GRPA or GRASP in Dictyostelium is required for the secretion of acbA. And because acbA is not secreted, you do not have SDF2. So this was therefore our handle to the physiology of the GRASP protein. Now, because it was secretion, we were excited because grass had been shown to not have any role in secretion, conventional secretion. Yeah? Now, I still remember the day when Matt Kinseth presented this data in our group meeting. And I looked at the sequence of acbA, and I said, my dear friend, acbA does not have a signal sequence. It cannot enter the endoplasmic reticulum Golgi pathway of secretion. How is this protein being secreted? Because in order for proteins to be secreted by George Pallady's pathway that we were all studying in the early 2000s, there is a strict requirement that the protein enters the endoplasmic reticulum. And for that, it requires a small peptide called the signal sequence. acbA does not have it. It therefore must be secreted by some strange mechanism. Yeah? And a little bit of research led us into the so-called, it wasn't called unconventional secretion at that time. So we learned that interleukin-1 beta, which was cloned in 1980s, late 80s, is released by cells, activated macrophages. And it also does not contain a signal sequence. And Walter Nickel was working on fibroblast growth factor 2, and that was also shown not to contain a signal sequence. So this therefore tricked us into thinking that here we have a new pathway to study. We can now address the business of how is it that cells secrete proteins that do not contain a signal sequence to enter the so-called conventional secretory pathway. So this was our entry in 2007 to this process of unconventional protein secretion. Now, so these proteins such as acbA, FGF2, interleukin-1 beta, and now there are many, numerous proteins. So insulin is secreted conventionally, and insulin-degrading enzymes are secreted unconventionally. Tissue transglutaminase, which modifies the ECM, which is something of great importance to us in our lab, is tissue transglutaminase is secreted unconventionally. Caspase-1 is now shown to be secreted by cells upon conditions of stress. Now, there are a few things you have to remember. Conventional secretion is going on all the time, whereas unconventional secretion is stress-induced. Okay? May not be all molecules secreted by this pathway, but vast majority are secreted only when there is an inflammation, oxidative stress, nutrient stress, or some other form of stress. Okay? Now, if these proteins are in the cytoplasm and they have to be secreted, there are many ways to go out. You could make a hole through the plasma membrane and go out, and you would learn more about that from Walter Nickel. Yeah? The alternative is, we had proposed a process called secretory autophagosomes. Yeah? We proposed in our 2007 paper that in the cytoplasm, upon conditions of nutrient starvation, which would activate autophagy, you would create an autophagosome which will capture a small amount of the cytoplasmic material. But this autophagosome, instead of fusing to the lysosome, would fuse either directly to the cell surface or perhaps an endosome, and you will end up with a multivesicular body or a vesicle secreted outside. Yeah? But we never found acbA in a vesicle outside the cells, meaning it was not the conventional exosome-mediated secretion. It was being released as a soluble protein. And we also found that acbA within the cells is contained in a membrane-bound compartment. It's very simple to do. You can collect total membranes, and if you just treat them with proteinase K, you find that the protein is protected, but if you treat it with proteinase K and detergent, the protein is lysed. Meaning, before acbA is secreted, it's captured into a small membrane, small or large, in a membrane-bound compartment. It's sealed in a way that proteases cannot access it. So the take-home message from this was very simple. acbA is picked up by a vesicle of some sort, and then this vesicle is processed by some other membrane of some sort and ultimately released. So what then is the mechanism and what then is the pathway? And this is where we have spent almost good old 19 to 20 odd years trying to address this issue. And I must admit, it hasn't been that straightforward. Yeah? But I think there's a lot of progress. So now, acbA is an acyl-CoA binding protein. There are many inside the cell. There are many different kinds of acyl-CoA binding protein. In fact, we've just published a paper that a gene called TANGO2 is an acyl-CoA binding protein. Yeah? But that's never secreted. It enters the mitochondria. But this acbA or acb1 is highly conserved. All cells have it. Its job is to bind acyl-CoA, but what is it doing outside the cells? Well, it turns out many things have been said. One thing we know for sure, its other name is diazepam binding inhibitor. So it does the opposite of Valium. In fact, this has been prescribed for schizophrenia in the past. Yeah? And there are many other things it does, but a more recent fashionable statement about acbA or acb1 is that it is linked to feeding behavior. So it turns out anorexic patients do not secrete acb1, and they haven't, therefore, the need or the urge to eat. So you can imagine then the value of knowing how this particular molecule is secreted because of its role in physiology. Yeah? So we worked in dictyostelium, but dictyostelium is not particularly an easy system to work with. So we decided to switch to Saccharomyces cerevisiae. And the problem with Saccharomyces cerevisiae is that you have the genetics, yeah? But it has a cell wall which makes life very difficult because these proteins are released in between the space between the plasma membrane and the cell wall. So this is the space where acb1, which is the yeast ortholog of acbA, would be released. But you have the cell wall. So how do you collect this material to quantitate it? In order to do that, you have to break this cell wall. And when you break the cell wall by using detergents or mild lysis conditions, you always end up breaking the plasma membrane. If you do that, then you would have the release of the cytoplasmic material. And then you would not be studying secretion. You would be studying cell leakage, which is not secretion. So Amy Kervin in my lab, trust me, please, if you would, spent almost 2 years trying different conditions to find one that would allow us to record acb1 secretion but not have any release of the cytoplasmic content, which should not be secreted. So cofilin and acb1 are present in roughly the same numbers in a yeast cells. They have roughly the same molecular weight. This is secreted upon starvation. This one is not. And we could therefore do the experiment in such a way that we could quantitate the amount of acb1 released by these cells. Now, the first experiment, of course, one would do with this kind of analysis is to ask, is GRASP required for this process? And when you delete the GRH1, which is the GRPA ortholog of GRASP in yeast, when you delete it, if you just, this is the same quantitated here, there's about an 80% reduction in the amount of acb1 secreted. Okay? You might ask, why don't we see a total loss? I don't know that. Okay? But 80% is good enough for us to work with in terms of trying to understand the mechanism. Now, it is very important to learn, to appreciate one aspect of this pathway of secretion, that we found that acb1-like molecules, and there are many others, they are secreted in one shot. So we find that if you start starving the cells, they don't secrete anything, but then by about 2 hours of starvation, there's a massive release of acb1, and then it plateaus. And if you don't have GRH1, of course, you don't secrete anything. What this tells you, there are 2 things to remember here. The cells secrete this as one bolus. Oop, off you go. Do whatever you have to do outside. And second, this rules out the possibility of any cell leakage, because if there was cell leakage, you will see a continuous increase in the release of the material over time. It plateaus. So about 90% of the protein remains inside the cells. Only about very low amounts is secreted by this particular pathway. Okay? So this was very important for us in terms of feeling confident that we are not studying leakage, we really are studying secretion. And of course, when you do these kinds of experiments, the obvious thing to do is to collect the total material and give it to the mass spectrometry facility of our institute and tell them to identify all the other molecules. Under conditions of nutrient starvation, we found that these cells secrete almost all of their so-called antioxidant protein. SOD1, acbA, I forget what this stands for, thyroidoxin 1, thyroidoxin 2, and another antioxidant. Okay? And it turns out, I will come to this later, but in mammalian cells, we can now reconstitute the same thing. And in fact, by UV irradiation, we can show that they also dump antioxidants. Yeah? So this is providing us a handle on a class of molecules that are being released by these cells in response to a certain form of stress. And I'm sure that this class of molecules vary depending on the stress applied to the cell type. Again, in yeast, we found that acbA here is contained within a membrane-bound compartment before it is secreted, just like we had shown for Dictyostelium discoideum. Which then brings us to the big question, what is this membrane-bound compartment? Remember, I told you that ER and Golgi per se are not involved. So what is involved? Where do these membranes come from? What is the pathway of acbA secretion? And because we had only GRH1 to work with, we tagged GRH1 with GFP, and we visualized its location in growing conditions and in conditions of nutrient starvation. So in growing conditions, GRH1 is found on Golgi membranes, and Golgi membranes in yeast do not look like Golgi membranes in the mammalian system. They are seen as dispersed elements spread throughout the cytoplasm, and there can be anything from 6 to 8 of these distinct spots. When you starve the cells, within 30 minutes you see coalescence of GRH1 GFP into 1 or 2 spots per cell. Okay? When you go from starvation back to growth conditions, this GRH1 is now found back on the so-called Golgi-like structures. So the question is, what is this material? Okay? We found that this material contains another protein that seems to interact with GRASP. It's called USO1, perfect co-localization. It contains one of the T snares. Remember, T snares are proteins that are required for fusion of membranes. Yeast Golgi has only one. It's called SED5. And SED5 is contained in some of the cups, but not all of the cups. A Golgi enzyme, so Golgi is known as a Golgi apparatus based on its organization of these stacks of cisternae, and because of the fact they contain Golgi-specific glycosyl transferases. Cups do not contain Golgi enzymes. So what you've done is you've taken Golgi membranes and partitioned them into another structure which contains some part of the Golgi but do not contain Golgi enzymes. Now, to summarize, because we have very little time, we found that phosphoinositides PI3P and PI4P were required for CUPS biogenesis and for secretion. COP1 and COP2 are the predominant proteins in a cell, are the most important proteins in a cell that are required for the formation of COP1 vesicles at the Golgi and COP2 vesicles at the endoplasmic reticulum. They are not required for CUPS biogenesis and for unconventional secretion. Escorts are proteins identified by Scott Emmer and colleagues, and they're required for the formation of intraluminal vesicles in endosomes. These are multivesicular bodies. So we found that escort 1, 2, and 3 were required, but more importantly, VPS4, which is the protein that is absolutely necessary for cutting a vesicle on the luminal face of an endosome, is not required. So we could therefore rule out the possibility of any exosome-like structures involved in the secretion of acbA1, even though escorts were involved in this process. Now, just to show you, we did some very fancy microscopy. Oops, sorry, let me go back. And you will see what we think cups look like. So we call this compartment that contains GRH1 cups for compartment for unconventional protein secretion. It's basically, it looks very much like a bicycle helmet. So if you cycle a lot, you would wear something similar to this, yeah? So it's it's a connection of tubules and small vesicles in this kind of organization, okay? So this is our so-called structure that is formed in cells when they are starved, and we believe this is the compartment. And we, of course, showed that acbA is contained within these cup-like structures. Okay? So to summarize this part of the storyline, GRH1 is found in membranes that are derived from the early Golgi membranes in yeast. We showed that they first assemble into a structure called pre-cups, and this is rather unstable, yeah? And they do not contain the cargo acbA. If you wait 2 to 2.5 hours, this is when secretion is observed. Then we found that the cups are more mature, more stable, and at this stage we could localize acbA to these compartments. Okay? Now, I told you that PI4P, phosphatidylinositol 4-phosphate, was required for cups formation and for secretion. Interestingly, the effectors of PI4P, clathrin and giga family of proteins, are not required. So what then is the effector of PI4P in this pathway? So again, this paper just came out in JCB last week, okay? So this then led us to look for the PI4P effector, and whether later part of Golgi membranes play a role in the so-called unconventional secretion. Okay? So again, to summarize, what we found was that DRS2, this is a very interesting protein, and I'll describe this shortly to you. It's found in the late Golgi. GRH1 is found in the early Golgi. Cups form from early Golgi. We found that DRS2 of the late Golgi, upon conditions of starvation, sits very close to cups. Cups here are shown in red, and DRS2 is in green. And sometimes they show very nice overlap, meaning almost becoming miscible, part of each other. Yeah. So, again, I don't want to go into the details. This is just published, so if you're interested in details, you can look it up. What we found that DRS2 has many effectors, but one of them called RCY1. We could test this easily. When you deplete RCY1, you do not secrete acbA1, you do not secrete SOD1, and you do not secrete thyroidoxin. These are the quantitations. This is the levels of defect. Similarly, we found that two proteins called V-snares, SNCC1 and SNCC2, these are proteins present on vesicles that allow vesicles to fuse to the target membranes. They are also required for this particular process. So based on a lot of analysis of this kind, we came up with this idea, proposal, that from the late Golgi, you make a compartment which we simply call modified TGN. I wanted to call this compartment teacups, but reviewer kept telling me, no, you cannot, okay? So this goes back to my storyline on tango genes, and I desperately want to call them tacos, but the reviewer said you cannot, so we call them tango. It still has a bit of Latin touch to it, yeah? I wanted to call them teacups because they really do look like, you know, if you grew up in England, you have your tea and then you put a tea cozy on top, and, you know, but anyway, it didn't work out. Modified TGN. Again, what we have is a compartment derived from the late Golgi, from TGN. It doesn't contain any of the Golgi enzymes. It contains DRS2, it contains SNCC2, and it contains TLG2. This told us right away that in the process of unconventional secretion, there is a lot of membrane fusion, budding and fusion going on, just like as in conventional pathway, okay? Now, we captured, so this is the work done with Aki Nakano. And unfortunately, it breaks my heart to tell you this, Kazuo, who worked on this project for almost 2 years with us, poor fellow died of pancreatic cancer, and he could not see the publication of this paper. He dedicated his life to this work. He did so much of this very fancy super resolution microscopy that Aki calls SCLIM. It's 4D. I don't even understand the details of it, by the way. He captured something really peculiar. He found that modified TGN in green is colored by cups, as shown in magenta, and you see a tube being, a tube coming out of this green cups, sorry, this green modified TGN. It's colored by cups, and ultimately this tube is cut. This is shown more clearly from a different angle. So this would be modified TGN, this is cups, and this is the coloring of this particular material by cups, and you see this gets cut here. What does this really mean? This reminded me of the old work of people working to show that ER and mitochondria, our mitochondria is cut through the function of ER by a similar mechanism, where a compartment is encircled by another compartment and something happens in this region and it undergoes cutting. So how would this happen? And this is work in progress. So DRS2 is a very peculiar protein because it flips phosphatidylserine, PS. Now PS is usually always on the luminal surface of membranes. It would be on the cytoplasmic face of the plasma membrane. But when the cells become apoptotic, one of the key signals is to flip phosphatidylserine to the outer face of the plasma membrane. And then those membranes bleb and they break. And that's a sign of apoptosis. So we know that DRS2 is required for secretion. Okay? So we have come up with the following scheme that in the modified TGN, through the function of DRS2, phosphatidylserine is flipped from the lumen to the cytoplasmic face. This then blebs. So remember, to create this intermediate, a transport carrier, we don't have COP1, we don't have COP2, we do not have clathrin, we do not have dynamin, we do not have gigaproteins. How do you create a structure which will then undergo cutting? How would you cut it? So we propose that it undergoes a process of blebbing. This, at some stage, recruits cups which color, and this color then cuts this structure. And we believe this is mediated by escort family of proteins because we have seen in our experiments and published that SNF7 is located at the junction of cups and the modified TGN, but it's transient. It comes late. So this, we propose, is the mechanism by which you create a tubule, which we haven't shown it yet, but will be filled with the cargo, acbA in this case, which would ultimately, this material will go to the plasma membrane, fuse, and deliver the cargo into the extracellular space. Okay? So how do we envision this to happen? So this is the, from 1974 to 2024, that's how many years? 50 years. The image of a conventional secretory pathway has changed. It's not simply that you form COP2 vesicles that take the cargo forward. Now, I think there is still a little bit of a concern even about the existence of the COP2 vesicles in the secretory pathway. I believe that COP2 vesicles exist. They're required for secretion of small molecules, but in the conventional secretory pathway, you have a new mechanism which mostly our work, we defined that through the function of tango-like proteins, you create these tunnels whereby you linked ER exit site to ergic membranes, and this large tubule is then cut, which becomes the foundational cisterna of the Golgi, and this matures into Golgi cisterna, so on and so forth, okay? So this pathway is totally new, yeah? But it is still very similar to what George Palade had said. You have ER, you have Golgi, okay? How you get to the Golgi has been changed. It's not just vesicles, it's also direct connection between compartments. In the unconventional secretion, so this is under normal conditions. When you stress these cells, these Golgi membranes now produce two kinds of structures, cups and modified TGN. 2 minutes and then I'm done, promise. These cups somehow collect cargo. This is where we suffer. We haven't a very good understanding. I think you're going to hear from Gail Young and Min of how cargoes likely enter this pathway. Whether it's translocation by a translocase or a vesicle fusing directly to this compartment, this is something we are actively trying to understand. But the cargo gets in here, it is then transferred to the modified TGN, which then creates a tubule or a vesicle. By the functions of DRS2 family of proteins, you create vesicles which fuse to the cell surface, thereby releasing a soluble protein to the extracellular space. So it's very similar in its overall features to the conventional pathway of secretion. But there are differences, as you can see. So I think this is building. This is, if I could say this, it's similar to what George Palade must have thought in the 60s. How is it that the conventional secretory pathway works, okay? We have ways to go, but we're getting there. Yeah? Now, quickly, we've also looked at, we've been able to reconstitute all of this process in macrophages. We work with macrophages because they secrete interleukin-1 beta, and they secrete something called MIF, which is the mother of chemokines, okay? So we have been able to show that we can trigger the production of IL-1 beta in these cells. So these are produced only when the cells are activated by LPS, okay? And after overnight treatment with LPS, you produce IL-1 beta, and then in the presence of ATP, you activate inflammasome and caspase-1, and you produce mature IL-1 beta, and it is this form that is secreted. And we can measure that. And to cut a very long story short, these are the pathways, and the take-home message here is that GRASP55, if you do not have it, mature IL-1 beta is not secreted. And what happens to that molecule is it gets aggregated, and it's degraded. So GRASP55 plays a very important role in IL-1 beta secretion. And if you don't have it, then the protein is not secreted, and it's degraded, hinting to the function of GRASP as some kind of specialized chaperone. Okay? So in summary then, there are elements that are conserved in unconventional secretion. You need 55 and 65 for unconventional secretion. 55, we believe, is linked to immune response. In another paper that just came out a couple of weeks ago, we showed that GRASP55 in dendritic cells is required for antigen presentation, which is again a problem of the immune cells, okay? And GRH1 is similar to 65, and we believe this is linked to nutrient and oxidative stress-induced secretion, yeah? So how these proteins serve in these two different processes is something worth addressing, and we are doing that. CUPS and a modified TGN are generated also in mammalian cells during starvation and in conditions of inflammation, for example, LPS. And we are trying to understand the similarities between CUPS and modified TGN in yeast versus the mammalian system. And with that, I stop, and these are the current lab members. The work that I've presented to you has been mostly done by Amy Kerwin, who has left the lab to start her own life independently, and is currently being worked by Prashna, who is here, Shomu, who is here, Natalie, who is here, and Jose Wainaki, who is here. And these are our funding agencies. And with that, I will stop and answer any easy, simple questions. Thank you. Thierry, yes. We need a global for this. Yeah. So we've done so many things. So, you are going to hear from Gail Young about TMED family of proteins. We've deleted all of them in yeast, and there doesn't seem to be any effect, right? There are 10 or 8 in yeast, or I can't remember how many. So we are struggling whether this is a special translocase of some sort, yeah? Or I still like to think that it is, so when viruses like HCV buds, it buds into the Golgi. It buds literally into the Golgi. So we think it might be something similar to that. Because if you simply grab material from the cytoplasm, you can't secrete it. You would be secreting ribosomes. You need to sort, and the whole function of conventional secretion post-ER is sorting. That's what you use Golgi for, sorting. And so we think the cups and modified TGN are doing just that, sorting. But how you enter these compartments, I can only guess. We don't really have an answer. And it's not that I'm hiding. We've tried, Julien is here. I don't know if he remembers our struggles in the lab, when he was in the lab. And so I don't know. I think translocation is probably the easiest to comprehend at this stage. But that comes with its own sets of problems. Yeah.
Hi, so I'm going to just ask one easy question. So you mentioned that only less than 10% of the acbA actually is it's a secreted.
I must change that to a little bit because in mammalian cells it's much higher.
Yeah, but anyways, it's in slide mode. So you show that it's secreted in a burst, right? Like after around 5 hours, it's all reached the plateau. But how could the cell control that? Since still 90% of those are left in the cell, why it's not secreting at all?
I don't know. So I think this is telling you something very important. It's a stress response. You release and you say, do whatever you have to, and that's it. Done. It's the same thing with cytokines. You don't just go on secreting. So I think it's physiologically very relevant. This is akin to what happens to synaptic vesicles. You release in one burst neurotransmitters. You don't go on dribbling. Yeah? So I think it's something similar to that. But physiology of that in a yeast or in a tissue culture cell is very hard to do. We would like to be able to do this in an animal model. So if somebody could do this in the real cells where we want to study this are astrocytes. That's where you secrete DBI. That's where you want to look at this because that's where you become sleepy or you become schizoid. So there's a physiological readout. Technically challenging, but that's where we are trying to go.
Okay, thank you.
Okay. Vivek?
Yeah, there you are.
Can you hear me?
Yes.
Great talk as usual.
Well.
Do you think how GRASP, the GRH is involved in the acbA and secretion? Does it have a direct role in COPS membrane generation, or is it simply a cargo receptor?
Well, Antonio is sitting next to you. He's going to present data, and I don't want to talk about his work, but one possibility is that GRASP acts as a sponge and accumulates acbA1 and then permits that acbA1 to be transferred into the lumen of the Golgi. Yeah? How? I don't know. But this is something that I also like, and we have some evidence of that GRASP really, 65, but 55 doesn't work like that, but 65 does. It acts as a sponge. Other than that, I seriously don't have any idea, but when we look at what GRASP binds, it just binds more chaperones, more and more chaperones. So we haven't found anything in terms of a specific molecule. And we think that might be limiting us because we might be dealing with an immunoprecipitation artifact under conditions of starvation. So
you don't think it is not related to the structure generation of the COPS membrane formation?
No, it has nothing to do with COPS, which then raises an issue. How do you cut Golgi membranes to create this new structure? Yeah, and we don't know that.
Vivek, I have a question regarding the cargos, right? So you mentioned there are multiple cargo proteins. Are there any similarities that you can spot between them? Like is that physical property or anything?
So when we do nutrient starvation, we find that they tend to secrete mostly antioxidants. Yeah. So in SOD1, this is the SOD1 that is mutated in Alzheimer's, for example. Yeah. Right. It's that form that is secreted. But it's mostly antioxidants, thyroidoxin, peroxyrhodoxin, et cetera, et cetera. We are trying to do that. That was done in yeast. We are trying to do that in the mammalian system, especially in macrophages. When they are stimulated with LPS, they secrete bucket loads of chemokines and cytokines. The question is whether there is something common. And the only thing I know we are going to find common is that they are cytoplasmic proteins. We mapped to two residues, two diacidic residues that are necessary for those cargoes to be secreted. But two diacidic residues is too little to act as a signal. So we play it down a little bit.
Thank you. Hi, hey Vivek, a very good talk. So I'm just curious about the first story when you showed that under starvation you all go through the unconventional secretion. Mm-hmm. But have you ever checked the conventional secretion? Yeah. Did they totally blocked?
Yeah, so when you go into unconventional secretory mode, the conventional secretion is highly reduced. It's not shut down completely, but it's virtually not. So in yeast, BGL2 is a protein that is inserted into the plasma membrane. When you go into the starvation mode, BGL2 is hardly there, yeah? And you're now secreting by the other pathway.
Thank you.
We haven't done that in mammalian cells.
Yan.
Hey, Vivek.
Hi.
Glad you say that there is some kind of role for GRAS proteins in Golgi. We don't have to,
yes.
Love each other, but we make conclusion based on our own, results, right? So my question here is, what's the role of GRAS proteins in making this structure?
Well, this is exactly what Mingu had asked me, that is it required for the creation of these structures? And I don't think so. I think GRASP is somehow just arriving as, I don't know, where is Gelyang? He disappeared. Well, there are people who think that CUPS might be an ERGIC-like compartment. Yeah, so it's basically moving from early Golgi and is now presenting itself on a compartment which is a combination, it's a collection of tubules and vesicles, which is ERGIC. And I think this is where Gelyang and I agree that this is something similar to a kind of, perhaps potentially an ERGIC-like compartment. But GRASP has no role I don't think I can...
How do you exclude the Golgi enzymes?
Huh? How do you exclude the Golgi? This is a big question. So this is telling you that there is a mechanism by which you can extract Golgi membranes and leave the Golgi enzymes behind. But it is not COP1. It's not COP1 derived vesicles that are creating this structure. And if you follow Ben Glick's work, and if you follow the work of many others, now there are many different kinds of vesicles. There's ATG9 vesicles. There's a vesicle that Tom Rappaport has talked about, which is created from the ER. It doesn't use COP2 coats at all. So I think there's going to turn out to be many, 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 many different kinds of vesicles that bud at the Golgi. Some use COP1, some don't. And in fact, I wouldn't be surprised that you have a coat-independent vesicle creation. Yeah. So that remains unclear. But when we look at these 4D images of Kazuo, and they are in the paper with lots of videos, you see that there are lots of vesicles that are coated with GRH1. They're coming together, they're coming together, then they fuse, then they create a bigger structure. What's in those containers is not clear to us.
Okay, for time's sake, let's last question.
Yeah. It's very nice cell biological and biochemical approaches, but I wonder, have you done some unbiased proteomic approach, for example, comparing the wild type and the knockout of GRASP the 55 to measure the secretome to see what proteins?
So we've done that in yeast, and we were doing it in the mammalian system. So we should have the data soon. Yeah, we've also purified the so-called cups, and we should have a proteome of that as well, hopefully shortly.
Right. Let's thank Vivek for this very exciting talk. And now I'm going to hand the